This is a brief review of heme metabolism for step 1 to get you ready for step 1. And this is to remind you of the blood cells, primarily erythrocytes. Red blood cells are the primary source, use a lot of hemoglobin, and therefore they have a lot of heme. And when the red blood cells die, that heme has to be degraded away and taken care of. We'll talk about that, as well as synthesis of heme for the red blood cells and for other purposes. Heme is used for hemoglobin. It's also used for myoglobin. It's also used for cytochromes. Of course, it's found in the erythrocytes, in the hemoglobin of the erythrocytes, and myoglobin. It's found in the cytochromes, in mitochondria, where we do oxidative phosphorylation, and for drug metabolism in the liver. So heme is an extremely important molecule that we've used for various purposes. So here's the structure of porphyrin 9, and the heme molecule is here with iron, ferrous iron added. It is in the reduced state, Fe+2, and that makes heme. So we take porphyrin 9 plus iron makes heme. And this is the synthesis pathway for heme. And there are a number of diseases associated with this called porphyrias, and they are defects in heme synthesis, and they can affect hemoglobin, myoglobin, and cytochromes, since they all contain heme. Some more different than others. We'll talk a little bit about that. But the synthesis pathway starts out in the mitochondria with succinyl-CoA and glycine, which are combined to make delta aminolevulinic acid, or ALA. Then that molecule goes out into the cytosol, where it's acted on to form porphobilinogen. And then porphobilinogen goes on to uroporphyrinogen 3, and uroporphyrinogen 3 goes on to porphyrin 9, and then iron's added in to make heme. Now, the heme controls this synthesis of more heme. When there's adequate heme, then it slows the synthesis down. So that's one of the feedback inhibition things you should see. Lead poisoning is very important in the heme metabolic pathway. There are 2 steps that are very sensitive to lead poisoning, and the first one is the conversion of ALA to porphobilinogen. This is inhibited, non-competitive inhibition by lead. The other one is the ferroketolase that adds iron at the very last step to convert porphyrin 9 into heme. So 2 steps, and these are both non-competitive inhibitions where the lead binds to a non-active site position on the enzyme. Now, the porphyrias. The first one we come to here is acute intermittent porphyria, and it's an acute disease caused by deficiency of hydroxymethylbilane synthase. And so porphobilinogen and ALA accumulate in the urine. The urine will turn dark on exposure to lighter air, and patients are not photosensitive. That's why I have the sun here. They don't mind the sunlight. The other form that I want you to focus on is porphyria cutanea tarda. It is a chronic disease caused by a deficiency of uroporphyrinogen decarboxylase, and uroporphyrinogen accumulates in the urine. So uroporphyrinogen 3 is the product that accumulates in the urine. It is the most common porphyria, about 1 in 10,000, and these patients are very photosensitive. Therefore, they don't like the light, and they tend to go out at night if they go outside. This one is both a liver and a bone disease. However, the acute intermittent is a liver disease. These people, the porphyria cutanea tarda, these people are thought to be how the terms werewolves and vampires were associated with some of these people early on. It's not a nice disease, but and sometimes people were made fun of because of that. Now, acute intermittent porphyria tends to be more of a spontaneous thing that happens, a mutation that occurs spontaneously, and these people Most of them, there's a small percentage that's inherited, but these people, it's mainly a liver disease, and so their disease is particularly exacerbated by taking drugs and alcohol, and they have neurological effects. King George III and Vincent van Gogh are thought to have had those. Now there are other porphyrias that you can see on this handout. I don't think you'll probably need to know them extensively. I think if you know these two real well and understand lead poisoning, you should be okay. Portoporphyrin 9 synthesis. Again, I said it starts out in the mitochondria, uses glycine and succinyl-CoA, two very common products, plenty of them. There are two aminolevulonic acid synthetase enzymes, ALS1 and ALS2. ALS1 is found in the liver and the bone. ALS2 is only in the bone. Both of them use pyridoxal phosphate or vitamin B6 to do this process, and they produce CO2 as a byproduct to make delta levulinic acid. And this ALA dehydratase is a zinc-requiring enzyme. It's found in the cytosol, and it's inhibited by lead. When acute intermittent porphyria occurs, you build up this porphobilinogen, and porphobilinogen is not a conjugated ring system. That's why these people don't have any problem with light. However, in porphyria cutanea tarda, you wind up with a conjugated ring system, not too different from portoporphyrin 9, and this absorbs light and causes blistering on the skin and red spots, and so these people don't like the sun. This stuff will deposit even in their teeth, and they will glow under UV light because of that. So those are various things to remember about these diseases. We'll talk a little bit more about sideroblastic anemia in a minute. That's a defect in ALS2. So the acute intermittent porphyria versus porphyria cutanea tarda. Remember, acute intermittent is liver, ALA, and porphobilinogen in the urine, not photosensitive. They like the sun. Drugs and alcohol exacerbate it. Neurologic and psychiatric problems, seizures and pain. Remember Mad King George III and Vincent van Gogh. Cutanea tarda is a chronic and the most common. It involves liver and bone. The product that builds up is uroporphyrinogen, and you see that in the urine. These people are photosensitive. They don't like the light. They only try to only go out at night. They were thought to be vampires or werewolves. Their urine will turn a very dark purple upon standing in the light. Sideroblastic anemia is defective due to ALS2. An immature ALS2 is used to make red blood cell, heme, nucleated red blood cells with ferritin iron granules that accumulate within the mitochondria. The granules often form a ring around the nucleus, but granules can be seen in erythrocytes as well. They are seen in peripheral blood smears. Thanks to Dr. Monaco for this information. There's slides on the structures here. The final step is in heme synthesis, goes back in the mitochondria, and that's where ferroketalates, also sensitive to non-competitive inhibition by lead, adds iron in the reduced state to the heme to make heme. Lead poisoning. The second step, ALA dehydratase, told you was sensitive to lead as the ferroketalase step, the last one. This results in a microcytic anemia, smaller than normal red blood cells. You see these red blood cells are small relative to this lymphocyte, and they're hypochromic. They look like donuts instead of having enough heat. They don't have enough heme in there, so the light shines through them. Quite often on step one, there's a question about an anemic child living in a home with lead paint. More recently, you may have a question from Flint, Michigan, where they've had a lot of lead in the water. This is an overview of heme degradation. When the red blood cells die, they're taken up by tissue macrophages in the spleen and liver, and these macrophages then degrade the globin part of the heme, and they release hemoglobin as bilirubin. Then the bilirubin is then transported to the liver bound to albumin. And once it gets to the liver, then it is conjugated. So it's transported unconjugated, just bound to albumin. Once it gets to the liver, it's taken up by the liver and conjugated with glucuronic acid. Then that goes out in the gallbladder into the intestines, where the conjugated bilirubin is secreted in the bile and then the intestine. In the intestine, glucuronic acid is removed by bacteria, and the resulting bilirubin is converted to urobilinogen, which is colorless. Some of the urobilinogen is then reabsorbed back into the bloodstream and goes to the kidney. And in the kidney, it gets converted into urobilin, which is yellow. The rest of the urobilinogen gets oxidized by bacteria in the gut to make it into stercobilin, which is brown. So feces are brown because of that, and urine is yellow because of what goes back into the kidneys. Conjugation with sugars. So it starts out in liver macrophage. The first product is actually biliverdin. You probably don't really need to know that, but biliverdin is also one of the things you produce from that is when it's breaks up that heme group, is to release Fe+3, and we have proteins that bind that and save the iron. And then carbon monoxide, which is toxic to hemoglobin, is released as well. There's very little CO released, so it's not really a major problem, but this is the one place in the body where carbon monoxide is produced. Then the biliverdin is converted into bilirubin, which is a more red-orange product. We wind up about 300 milligrams of that a day, and it's not water-soluble. So it can be bound to albumin's hydrophobic regions, gets taken to the liver, where it's conjugated or linked to diglucuronide using UDP, glucuronic acid, and then the conjugated form becomes water-soluble and can then be secreted. It goes into the bile and handled. Jaundice. When jaundice occurs, we have yellow eyes and yellow under our tongue and various other places. Skin. When it's really severe, you'll have yellow skin. There are three different forms of jaundice. There's hemolytic jaundice, where red blood cells, hemolysis, RBCs are dying faster than heme can be degraded. Hepatocellular jaundice, liver damage, such as in hepatitis or cirrhosis of the liver. Liver can't conjugate bilirubin fast enough. And then obstructive jaundice, where you have a bile duct obstruction. You might have gallstones or pancreatic cancer, pressure on the bile duct that results in it being stopped up, and so the bile can't get out of the liver. In that case, the conjugated bilirubin builds up in the blood. In these other cases, you don't have any. You have low levels of conjugated bilirubin and high levels of unconjugated bilirubin. Urobilin secretion in the urine. The gut bacteria convert conjugated bilirubin to urobilinogen, which is colorless. Some is reabsorbed. Urobilinogen is transported to the kidney and converted to yellow urobilin. The rest of the urobilinogen is oxidized by intestinal bacteria to brown stercobilin. Jaundice of the newborns. Young, immature, premature babies, quite often their liver is not mature enough to do the conjugation step. They lack the adequate amounts of UDP, glucuronyl transferase, which is the enzyme that puts the glucuronic acid onto the bilirubin. Consequently, they could treat these kids with UV light that oxidizes the bilirubin to a more water-soluble product. And so this just shows you the data from normal, full-term, and preterm premature babies. Bilirubin measurements. There's conjugated bilirubin or direct reacting. Now, the conjugated form is direct reacting, and it is water soluble. So it's the water soluble fraction of blood that has the bilirubin in that. The total is a mixture of conjugated and unconjugated, and it is a methanol soluble fraction. So the total minus the conjugated equals the unconjugated, and it's the unconjugated form that is not water soluble that gets deposited in the sclera of the eyes to cause jaundice. So again, here's an overview. Remember, it goes from the red blood cells, macrophage, liver, conjugation, bile duct, urobilinogen, back in to make urobilin, and then stercobilin, and then that's the final products there. Okay, thank you. Hope it helped you. Bye-bye.